Now here we're going to go over fermentation. Now, fermentation is an anaerobic process. Now, if we think back to glycolysis, within glycolysis, we start off with a glucose molecule and we oxidize that glucose molecule into two pyruvate molecules. Now, depending on the conditions in the cell, pyruvate can have three different fates. It will either be converted into ethanol, lactate, or it can be converted into CO2 and water. Now, in aerobic conditions, that pyruvate will get processed into acetyl-CoA, and then it will enter the Calvin cycle where it will be oxidized into carbon dioxide, and we'll see water as a product, and those electrons will get passed on to NADH and FADH2, which will then enter the electron transport chain. Now, in aerobic respiration, we will see a lot more energy production because that pyruvate is going on to the Calvin cycle and its electrons are going to go on to the electron transport chain. So overall, we see a lot more energy being produced because of that electron transport chain. But in anaerobic conditions, we do not see that much energy production. So then you may ask, what is the purpose of having an anaerobic process? And recall that anaerobic and aerobic, these are terms that describe the conditions of oxygen in the cell. Aerobic means there is oxygen, and anaerobic will mean a process that does not require oxygen. Now, anaerobic processes are important specifically in the topic of fermentation because they allow for NAD+ to be regenerated. So remember that in glycolysis, we see that NAD+ will get reduced to NADH, and this NADH will go on to the electron transport chain. But if we cannot go towards the Calvin cycle, and if we cannot go towards the electron transport chain cycle, how are we going to oxidize that NADH and bring back NAD+? So essentially, we get NADH, right, from glycolysis, but we need to oxidize it back to NAD+ so it can be used again in glycolysis. Because the cell has a limited amount of NAD+, so it must be replenished. Now to reiterate one more time, remember, glycolysis produces NADH. This NADH will get oxidized in the electron transport chain, but if it cannot, we need an alternative pathway to oxidize that NADH back into NAD+, and that is where we need these anaerobic processes. So in these anaerobic processes, we produce ethanol and lactate, but we regenerate the NAD+, which can be used in glycolysis. Now, the key thing we need to understand, fermentation is performed under anaerobic conditions to replenish NAD+ for glycolysis. That is its whole purpose. And then once that NAD+ is replenished, glycolysis will occur and it will be the main source of energy because remember, glycolysis produces a net yield of 2 ATP. That is a very small amount in comparison to aerobic respiration, which produces a significantly larger amount of ATP. Now moving forward, we're going to look at ethanol fermentation first. Now, in ethanol fermentation, remember, we start off with pyruvate. Pyruvate can have various fates. It can turn into ethanol, lactate, or CO2. Now, in ethanol fermentation, there are two steps. First, the pyruvate must get converted into acetaldehyde. Now, this reaction is catalyzed by pyruvate decarboxylase. Whenever we hear the word decarboxylase, I want you to think of removing a carbon dioxide group. Now we can see on pyruvate, we have this COO group right over here, which is going to be removed, and we are going to get our carbon dioxide. After that decarboxylation step, we will yield acetaldehyde, in which we will lose a carbon group. We went from a 3-carbon molecule to a 2-carbon molecule. Now in the next step, that is where we see the oxidation of NADH. Remember, we are trying to replenish the NAD+. Now we have this 2-carbon acetaldehyde group, and we are going to reduce this molecule into ethanol. So we're reducing acetaldehyde and we're oxidizing NADH. And as a product, we get reduced ethanol and we get oxidized NAD+. And this reaction is catalyzed by alcohol dehydrogenase. Now, to reiterate one more time, we start off with a 3-carbon molecule and we end up with a 2-carbon molecule. That first carbon dioxide is lost as CO2, so it's a decarboxylation step. And then we see an oxidation reduction step where acetaldehyde will get reduced to produce ethanol and our NAD+ is regenerated through the oxidation of NAD+. Now, the overall reaction can be depicted as so. So we start off with glucose, and remember that glucose must be phosphorylated, and in glycolysis, it's actually phosphorylated 2 times, in step 1 and in step 3. We need 2 ADPs because remember, glycolysis will produce 2 ATPs. And then we will produce 2 ethanols. Why are we producing 2 ethanols? That is because, remember, glucose, when it breaks down, glucose is a 6-carbon molecule. It will get broken down into 2 3-carbon molecules. And what are those 2 3-carbon molecules? That is pyruvate. So glucose gets broken down into 2 pyruvate molecules. And those 2 pyruvate molecules, when they undergo ethanol fermentation, they will produce 2 ethanols. And then we also get the production of 2 CO2s. Why? Because each round of ethanol fermentation will produce 2 carbon dioxides. And then we have 2 ATP. This 2 ATP is coming from glycolysis because remember, glycolysis has the net production of 2 ATP. And then we also have water as a byproduct. Now, one thing to make note of is that in this process of starting off from glucose and going to ethanol, there is no net oxidation or reduction. Now, I want you to think about this for a second. Why is there no oxidation reduction, no net oxidation reduction? Well, in the process, we are oxidizing and reducing, but overall, there is no net change because in glycolysis, what are we doing? In glycolysis, we start off with NAD+ and we convert it to NADH. This was a reduction, right? But then in fermentation, we take NADH and we convert it to NAD+. Now, this is an oxidation. So overall, there was no net change. We went from a reduction to an oxidation, no overall change. And that is essentially ethanol fermentation. And in another video, we will go over the production of lactate, which is also through fermentation.